Hello everyone, and hope that you're doing well. Today we're looking through this paper 2, CSEC biology paper. This biology paper is from May-June 2023. As always, please remember to read through the instructions carefully before you start the examination, and if you should have any questions or queries, clarify those with the examiner before you begin. Now let us jump into section A, question number 1. And this reads that figure 1 shows micrographs of an animal cell and a plant cell. And you'll notice carefully you see the animal cell on your left and the plant cell on your right. Now let's jump to the questions. Now the first question, part A, it said what type of microscope was used to produce the images shown in figure 1, and this is an electron microscope. How we know this is because of the details of each cell. All right, let's go to the next question, part 2 of A. It said draw a large, clearly labeled diagram of one plant cell from the micrograph shown in figure 1. All right, I've drawn my diagram here with the labelings. Let me just zoom in a little bit so you get a better scope of this diagram. All right, and here we go. So your diagram again must be large. All right, let's go one more time. Right, that is good. So your diagram should be large, relatively large, and properly labeled. And if you notice, you see the cell wall, you see the cell membrane, the nucleus, the nucleolus, the cytoplasm, the chloroplast, the vacuole, and the mitochondria. Again, this also proves that it is an electron microscope because of the details that are in the diagrams. All right, a light microscope, as you know, will not produce such detail. More likely, you will only get the outline of the cells. All right, so let's go to our next question, our part of question. All right, so let's go down. And this is part 3. All right, and this question reads now, it says list 2 similarities between plant cell and the animal cell. All right, let's go a little bit more in our Zoom. All right, just be a second. All right, that is better. All right, so similarities will include cell membrane, ribosomes, vacuole, cytoplasm, nucleus, and mitochondria. So both cells have these structures in common. For part 4, it said the most prominent structure on the animal cell electron micrograph carries genetic information in the form of DNA. State the name of this structure. And notice here it said that this is an electron micrograph. And this is another clue to prove that it is an electron microscope. All right. However, the prominent structure in the diagram, if you could go back, you'll notice that it is the nucleus that is shown. So definitely the nucleus is that structure that is most prominent on that cell that carry the DNA. All right. And of course, which I'm going to discuss this a little bit further down, but you should also know that chromosomes carry DNA. All right. But the chromosomes are not that obvious in this diagram unless the diagram is not clear. Otherwise, you can use chromosome as a structure as well. All right, for part B, so the diagram in figure 2 shows the general structure of a bacterial cell. All right, and here it asks to identify one similarity between the bacterial cell and the plant cell. No, the first question actually asks you to label this, I think. There's a question talk about labeling. All right, it's down further. All right, so we label the structures. I don't need to go back. I just want to make sure you actually understand and know the structures of the bacterial cell. We have our flagella. All right, flagellum for singular. We have pili, and pilum will be singular. We have our plasmid. We have our DNA. And you can actually label it as circular DNA. We have our ribosomes. We have our capsule, which is the outermost layer. We have our cell wall and cell membrane. So part 1 of this question asks us to identify one similarity between the bacterial cell and a plant cell. And the similarity between a plant and animal cell is that both bacterial cell and a plant cell, they have a cell wall. All right, so that is similarity between both. For part 2, it said the structures of the bacterial cell are especially suited for its role in infecting other organisms. Table 1 lists 4 of these structures. Complete table 1 by explaining how each structure helps the bacterium to infect organism. An example as being provided for you. All right, so we have the structures there. We have our pili, which is also called our fimbriae. We also have our flagellum. We have our slime capsule, or you can just say capsule, and we have our cell wall. All right, as they gave us the example up top there. So I'm going to go straight into flagellum, and flagellum enables movement towards the target cell or the host cell. All right, and the capsule will protect the bacterium from harmful chemicals and also help the bacterium to stick to the target cell. All right, and the cell wall also provides some form of protection as well, and it provides protection against antibodies and also the phagocyte. All right, now we are at part C, and it said human beings are multicellular, all right, and are made up of a large variety of cells. It's a complete table too by distinguishing between the following cells. I'm going to distinguish between red blood cells, skin cells, and nerve cells. And so the difference between red blood cells and skin cells is that the red blood cells have no nucleus, while of course the skin cell will have nucleus. All right, red blood cells are biconcave in shape, generally smaller than the skin cell, and contains hemoglobin, which is of course the skin cells. Skin cells have none of these. The difference between skin cells and nerve cells is that nerve cells have extensions and they are called axon and dendrites. Skin cells lack axon and dendrites. Again, nerve cells are insulated with what they call myelin sheath. All right, and of course, skin cells do not have those. Now, for part two, it's a name one feature that would enable a scientist to distinguish a spherical bacterial cell from an animal cell when viewed under a microscope. And one of the most obvious thing that will be noticeable is the bacterial cell or bacterial cells generally will lack nucleus because they do not have a nucleus or a true nucleus. Also, what is important is that the bacterial cells, they have one circular DNA, which I label up top. All right. Also, points that you can also discuss or include, depending on the number of marks. You can say the plasmid will be in the bacteria. The bacteria cells will have cell wall, they have a capsule, they have flagella, and also pili. All right. And of course, an animal cell will not have those. All right. All right. Question number 2. Figure 3 is an incomplete diagram of meiosis in an animal cell. It's a four genetically different daughter cells, P, Q, R, and S, are produced. And notice we have chromosome up top there. Then we have meiosis. And these will be our final cells, which are called daughter cells. And again, they are genetically different. Just to make a point right there, say we need to complete the diagram to show the difference. That's what part A is saying. So what I did now, if you notice what I did, just to make sure you understand this, notice what we started out with. We have a long dotted line or broken line chromosome. We have a short one that is broken line, and we also have a long solid line and a short solid line, which is more curved. All right. So we have helix shape chromosome, based on the diagram. So you notice the ones they put in P and S. Okay, and they put two solid one in P, two broken lines, one in S. So what I did now for Q and R, which again you can have a different varieties of these things. The most important thing, you can only have two chromosomes within each of these cells. Why? Because meiosis only produce half the number of chromosomes. That's the first thing you have to know. Now, what is also important, because of what they call crossing over and synapsis in prophase 1, what you're going to have is that you'll have sharing of genetic materials. So notice what I did is to mix the solid lines with the broken lines in different ways to get my Q and my R. All right? And of course, also what will be acceptable as well, if you put a solid one and a broken one as well, that will be also acceptable. But remember, is that sister chromosomes will separate, all right, to go to opposite pole. So hence you will not have the same or different chromosome within the same cell. What is also important too, remember that chromosomes are independently assorted as well, just to make mention of that. All right, what is important about the difference, just a notation right here that you need to remember, is that the differences in daughter cells for meiosis, it is due to the crossing over and synapsis in prophase 1. All right. Now, for part two, it's a state whether the daughter cells produced by meiosis, or you may pronounce it as meiosis, are haploid or diploid. And meiosis only produce haploid cells. Haploid means half the number of chromosomes. So, for example, sperm cells, right, and egg cells, they only have half the number of chromosomes, and when they fertilize each other, then what happens is that the numbers now come together to give you a full number, which we call diploid. All right. For part B, it's a name one part of the body in which meiosis occurs, and this may take place within the ovaries or testicles. So ovaries if you're a girl, and testicles if you're a boy. All right. Now for part C, it's a name the structure within the nucleus, which contains the genetic materials, and this will be chromosomes. Chromosomes contain the genetic materials. All right, and just a point to note right here again is that genes make up DNA, while DNA make up chromosome. All right, so chromosome will be the large molecule, and portion of the chromosome we call DNA. And then, and again, just to make note, is that when the DNA, when the chromosome uncoiled, all right, around histone protein, that's when they get DNA, and segments of DNA is what you call the genes. All right. All right, so just to make note of those extra information there. All right, so part D is said Kara and Keturah are daughters of Mr. and Mrs. Balaram, yet their observable characteristics are very different. It's a user knowledge of genetics to explain why the observable characteristics in the sisters are different. Include two possible reasons in your response. And I just give you the reasons with the explanation one time. And so for reason number one is that during meiosis, especially prophase 1, all right, we have crossing over result in homologous chromosomes exchanging genetic materials. And now this accounts for the differences in the daughter cells or gamete. All right, and for reason number two is that parents can be carriers of recessive alleles, hence the possibility of homozygous recessive genotype resulting in different phenotype compared to siblings with dominant alleles. So again, if they are carriers of the recessive alleles, that will not show up. Only when those recessive alleles come together, then you will see that characteristic. So you may see a sibling with certain characteristic, and then you see one with a different characteristic is because of the difference between dominant and recessive allele. All right, so one, it could be because of crossing over, exchange of genetic material, and another one is because of the differences in dominant alleles and the possession of recessive alleles. All right, so part E. State two reasons why meiosis is important to living organisms. And one of the most, I wouldn't say it is the most, but one of the most important thing is that it results in what they call genetic variation or diversity in organisms. And of course, we know why, because if organisms are all the same, then, for example, one particular disease could wipe out everything. You would have easy extinctions of organisms. So variety, variation, diversity will give rise to more sustainability of organisms. All right. And of course, the adaptation process will be better as well. Another option that you can put there is that because of meiosis, you can suppress or reduce genetic disorders. For example, sickle cell anemia, right, versus normal blood. All right. So of course, if a person with sickle cell mate with a normal blood, the child may not come out with sickle cell and eventually may fade out depending on the mating with normal blood persons. All right, so the next one is that increased adaptation and survival of organisms, and that one is closely related to number one as well. Number three is closely related to number one, and that is because of diversity and variation, of course. Another point here is that it can aid in the production of gametes for sexual reproduction, and another point is that provides means of maintaining diploid number of chromosomes when gametes are joined, because meiosis give you half. And so because you have that half, when they come together by means of fertilization, you get the full number. All right, so it's very important to do that. All right, part two, it stated that meiosis also occurs in flowering plants. It said name the two gametes produced, and those two gametes are pollen and ovules. And point to note is that each pollen has two sperm cells. All right. And I think there was a question one time that I think they give you like two pollen grains and ask you how many ovules can be fertilized. And all you need to do is to multiply that number by two, we get 4. All right. So things like that you need to just bear in mind when you're going through these papers. Question number 3 is a list 3 kingdoms other than the animal kingdom into which organisms can be classified. And I have 4 options to choose from. You have the plantae, which is plants. You have the bacteria, otherwise called the monaria. We have fungi and protista. All right. For part B, it says fish are organisms that belong to the animal kingdom. Is a figure 4 shows 6 species of fish, A to F, that live on coral reef in the in the Caribbean. All right. And so here you have the 6 of them. And here we have the 6 species. Notice they're very different, of course. And these are Caribbean fishes. And so for part 1, it said list 3 visible characteristics that may be used to classify the fish in figure 4. All right, so if you look at them, you notice that the shape and size of their fins are different, right? So you can put them in different groups based on size and shape of their fins. We can also look at the body shape and size as well. The body, not all the same shape and not all the same size. The type of shape of their mouth. The mouth is also different if you notice them as well. You have the patterns on the body as well. So patterns on body is also a difference. The markings or patterns or contour in some cases, they're all different, okay? All right, and so the next part of the question asks us now, so using any one of the characteristics listed in B1, arrange the fish species A to F into 2 groups. And we have group 1 and 2. And so they give us the 2 columns here. So group 1, and it's a characteristic used. So we're going to use large mouth compared to smaller mouth, which, and the smaller mouth ones, you notice they are typical shape of fish mouth, right? And if you look at the diagrams, you notice that fish C and E, they have larger mouth or unusually shaped mouth. Especially, I think was what? Let me just go back 1 second. Notice that C, like a snout type of mouth, a very long, unusual mouth. And E, which is a grouper, very large mouth as well. The others are typically small mouth. All right, so that's 1. You can also use the fin. You can also use the patterns and put them in different categories as well. So you could do anything to get those 4 marks real easily. All right, for part C, 1, it's a define the term conservation as used in biology. And there are some important words you want to put into this. And it is any process or method or attempt to manage, protect, and preserve natural resources. So you must use the word manage, protect, preserve. Those are very important words to use when it comes to conservation. All right. Now for part 2, it's a fish populations which live on coral reefs in the Caribbean are under a threat of extinction due to predation and pollution. All right. So it's explained 2 methods of conservation that may be used to minimize these threats. But point to note here before we go to these threats is that when we talk about predation, it's not only fish eating fish or larger sea animal eating fish, but we also hunt and capture fish as well. So we are their predators. That's what to bear it in mind. I know sometimes when you think about the marine ecosystem, you only think about, when you talk about predation, prey or predator, you refer to the larger organisms like shark and all those are bigger fishes. But definitely we are predators of fishes as well. All right, so 1, we can establish proper garbage disposal methods. So you don't pollute the water, of course. You can design proper sewage treatment plants to prevent sewage from entering the water body. All right, and that word is spelled incorrectly. I'm just quickly change that, entering. And a point to note is that as I do this quickly, I just wanted to note that when I think about sewage entering water body, you need to think about, I make an error. Anyway, all right, so the sewage enter in water body to prevent pollution. And again, what is important with that point that I made is that sewage will lead to what they call eutrophication, which is excess growth of algae. And just mentioned that. And next point here is that encourage the reduction of pesticides and fertilizer use among farmers near coastal areas. So the farmers that live near coastal areas, encourage them not to use pesticides and fertilizers and so on. All right. Again, there is missing from the fertilizer as well. Just bear it in mind. I'm not going to change it because I'm going to erase it. I don't know why. But anyhow, next point here is that establish and enforce open and closed season. And the reason for open and closed season is that the closed season will allow the marine organisms or the fish to reproduce, all right, give them chance to reproduce and replenish their population. You can also sanction the prohibition of releasing pet fish or other marine species in water bodies, and this will reduce the possibility of invasive species. All right, and lionfish is a problem at this point because the lionfish is an invasive species in the Caribbean, so definitely and I guess it was released maybe by a fish owner that no longer need that fish into the large body of water, and then it become invasive. All right, number 4, it said organisms can be either multicellular or unicellular. State one example of a unicellular organism, and a pretty much easy one here is bacteria. So that would be my first answer. We have other examples that you can also list, such as protist. And protist, example of that will be amoeba, paramecium, or euglena. All right, so those are some examples there. Yeast, for example, can be as a single cellular, and yeast will fall under fungi, but of course, not all fungi are unicellular, just to make mention of that. Now, part 2 is a list 3 materials that can be transported in multicellular organisms. And this, you can just run out on this one because we have so many things we can list. Water, for example, glucose, amino acids, mineral salts, hormones, fatty acids, glycerol, sucrose in plants, waste, example, urea. All right, so there are so many things that we can list for that one. And for part 3, is it explain why multicellular organisms require transport systems while unicellular organisms do not. Include 2 reasons in your response. And so what I do is to put the reasons and just explain one time. And so the first reason is that it's based upon size, okay? And that multicellular organisms are bigger or have lower surface to volume ratio. And so because of this, they need transport system to take substances to and from the many cells over relatively long distances. All right. Also, they need transport system because tissues are generally large and require a large supply of substances. So therefore, you have to transport these requirements of substances towards these tissues or cells. Reason number 2 is that exchange of substances directly between the external environment. And these are the points now to make note of, or the point to make note of. So unlike unicellular organisms, most cells in multicellular organisms, they are unable to exchange substances directly to the external environment. For example, your alveoli, your lungs, and then it will take oxygen towards the cells of the body for respiration. These cells are embedded deep within inside of you. So therefore, you have to transport the oxygen through your respiratory system. Motherfucker, it goes through your breathing system first, then it goes through your blood, and then it goes through those cells because those cells are deep within inside of you. A single cellular organism is just exposed directly to the environment. So they can just capture the gases that they need from the environment if they do need gases. All right? And point to note here is that, again, just the last sentence there says, hence transport systems are needed to effectively take substances to and from these cells. All right? Now, next question here, it said define the terms translocation and transpiration. And this paper is relatively easy. It asks a lot of definitions for some reason. Now, translocation is said that the transport of plant food through the plant by the phloem. And this plant food that I'm talking about is generally sucrose, okay? Sucrose, not glucose, sucrose, all right? Now, transpiration is evaporation of water. That's very important to mention. The transpiration is the evaporation of water from the uppermost parts of a plant. Generally, the leaves I'm talking about, right? All right, so part 2 here now, it said explain 2 ways in which the structure of the xylem is suited for its role in transpiration. Now, this question is kind of asked a certain way because looking for the importance of xylem in transpiration, not the importance of xylem in terms of transportation, but transpiration. All right, remember that xylem transports water and mineral salts, but how the xylem is suited to assist transpiration, which is the movement of water through the plants, and then it will evaporate through the leaves. So one is that the xylem is dead, is a dead tissue, and since it's dead, it allows water to travel through easily without being absorbed along the way. Because if the xylem cells or tissues were not dead, then they themselves will absorb water. So the xylem is like a straw, dead, hard structure, rigid. All right? All right. And so it is also narrow. And because of this narrowness, it aids in the capillary action. All right? That means water could climb through the xylem easily. All right. And so water can move upwards without any resistance or little resistance against gravity. All right. Also, it's tube-like, which is kind of related to narrowness, of course, but it's tube-like for the fact it's tube-like. And since it's tube-like, it also allows capillary action and easy passage of water. All right. Water travels through tubes quite easily compared to other stuff. All right. And again, to note, because of these factors or adaptation, we may call them, water can passively move upward through the xylem. So it moves easily without little to no energy, all right? Or little to no resistance. Now for part 3, it said suggest how the structure of the phloem allows it to deliver nutrients to sieve elements. And this is one simple process called diffusion. However, based on the nature of the concentration of substances, is actually active transport because the substances will go against a concentration gradient. All right, so you put diffusion and put active transport, then you get the answer. Or you can just put active transport is also fine. All right, but the major process is diffusion by active transport. All right, and so for question number 5, we're almost there. It said define the following terms. Again, this is loaded with definitions. I don't know, they might want to give away points that here. So it said diffusion is the first word to define, and diffusion is the movement of particles from an area of high concentration to an area of low concentration until the particles are evenly distributed. Breathing is the next term that they want us to define, and deep breathing is the process of inhalation. So inhaling, which is taking air in, and exhaling, which is the removal of air. All right, and the final word on this question to define is respiration. And respiration is the process by which nutrients such as carbohydrates, particularly glucose, are used to produce energy. So it's really the production of energy from food, all right? But I just want to make mention that that energy that food we're talking about is actually glucose. Carbohydrate is the primary source of energy, all right? All right. Now, next part of question here now is part B. It said name the 3 muscles in the rib cage which are responsible for breathing in humans. And typically, you'll have the diaphragm, you have intercostal muscles, and matter of fact, doesn't make mention here. You have the internal intercostal muscles and you have external intercostal muscles. And if you go to the CAPE level, you might learn about also inner intercostal muscles. So you have actually 3 intercostal muscles. But sufficingly for this question, diaphragm, internal intercostal, and external intercostal, they are sufficient for this level. All right, part C. And for part C, it said explain how the process of gaseous exchange occurs in the lungs. And I kind of give you a whole outline of how the air reaches the lungs first, and then I just mention it at the bottom. Will actually take place in the lungs. So the first thing that air is a mixture of gases. Just to make mention of that, it enters the nose or nostril and also the mouth, and air moves down the trachea All right, and the trachea is kept open by a C-shaped ring, rings of cartilage, a matter of fact, and then air then enter the branch off into the bronchi, and then air then enters the bronchioles. Once it in the bronchioles, what's going to happen here now is that air then enters the alveoli. And alveoli, those are the sac-like structures that are attached to the ends of the bronchioles. And what is important, the alveoli, they are surrounded by many capillaries. This is very important in the gaseous exchange process, by the way. Now, oxygen now is able to diffuse across the alveoli into the capillaries, while carbon dioxide will diffuse from the capillaries into the alveoli. And that is how we have the gases exchanging. So carbon dioxide is exchanged for oxygen, and carbon dioxide is exhaled out of the body, of course, in the reverse process, going towards the nostril and the mouth. All right? So again, in the lungs, we must talk about the capillaries, many capillaries around the alveoli, and the movement of the gas in and out. And what is also important for you to mention within the lungs is the concentration of these gases. So in the alveoli, what you're going to have, and please make a note of it, please write it down. So in the alveoli, what you're going to have is a higher concentration of oxygen. Therefore, it will move from the alveoli into the bloodstream, where there is a low concentration of oxygen. However, now in the capillaries surrounding the alveoli, you will have a high concentration of carbon dioxide. Hence, carbon dioxide will move into the alveoli, where there is a low concentration of carbon dioxide. And this process is diffusion. So you must mention that. Please mention that. Write down those. All right, I should have written down all of that, but space. All right, the next thing to make note of is that the alveoli must be thin, moist, with large surface area to be effective in gaseous exchange. So this bit of information, very important for you to understand the whole process of breathing and gaseous exchange. Now let's jump to the next part of the question here. Part D. It said, explain how human beings use anaerobic respiration during exercise. And a point to note that I'm just going to use this concept of explain oxygen debt. All right. And so during strenuous exercise, oxygen sometimes become low, okay, in the cells. And this is because oxygen is used up faster than it can breathe in. All right. However, though, even though oxygen is low, respiration still takes place. All right, so what happened in the muscles now? Because this is where anaerobic respiration takes place in the muscles. Point to note as well that in animals, including us, we undergo anaerobic respiration in our muscles, but particularly in the cytoplasm of the cells. Anaerobic respiration does not occur in the mitochondria, but it takes place in the cytoplasms of the cells. So please, cytoplasm of cells, that's where it is taking place. Please make a note. All right. So point to note here is that anaerobic respiration, which is called lactic acid fermentation, that's occurring in us, because you have another anaerobic respiration, which is called alcoholic fermentation, not in animals, but in microorganisms such as yeast. All right. So again, anaerobic respiration, which is lactic acid fermentation, occurs instead of aerobic respiration because of low oxygen. So without oxygen, glucose molecules breaks down into two lactic acid molecules. All right. And so half of a structure of a glucose is technically a lactic acid. They're not the same. It's not exactly equal to half because the structure is different and it is acidic in nature. All right. And so this releases a small amount of energy in a relatively fast time. So of course, during strenuous exercise, you need a quick energy. You're going to respire anaerobically to get that quick energy. However, it will not be a lot. What is also important during strenuous exercise, you produce a great deal of lactic acid, which will cause cramping or may cause cramping. All right. All right. So just to make note that the muscles rely on this quick release of energy to keep working. All right, and now we are on to our final question. Again, a definition. This paper is loaded with definitions. Now, it's a define the terms egestion and excretion. And egestion is the release of undigested food from the body, while excretion is removal of metabolic waste from the body. And metabolic means from chemical reactions. All right. And so for respiration, for example, is a metabolic process. So therefore, carbon dioxide is a waste. All right. Just to make mention of that. All right. And it said part B, it said in humans, several organs are responsible for excretion. All right. So list 2 organs of excretion, and for each organ, state one substance which is excreted from it. So, for example, the excretory organ, which is the lungs. All right. And the substance released from the lungs is carbon dioxide. All right. We also have, and remember that carbon dioxide is produced by the metabolic process of respiration. We also have kidneys, and kidneys will release urea and excess water. And urea comes from the process of deamination. It's a chemical process, so it's metabolic. Deamination is a chemical process where, of course, excess amino acids are converted into urea. There's a number of steps that take place. You can check out my a lesson that I have on deamination. All right. And skin will release urea as a waste, metabolic waste, and also excess water as well. All right. And so for part C, is a Joy is ill and is vomiting a lot during the day. She becomes very dehydrated as a result of her condition because she has lost a large volume of fluid. Explain the role of the kidneys in correcting Joy's fluid imbalance. And a first point to note is that as she becomes dehydrated, her blood becomes concentrated, which means less water, not dilute, but concentrated, all right, with every other substance except water, all right, because water is a large part of the blood. Now, the second thing will happen here is that this signals the secretion of what they call ADH, which is antidiuretic hormone. And what's going to happen here is that more water is reabsorbed because of ADH. More water will be reabsorbed in the nephrons, which is the tubules of the kidneys, particularly the proximal convolution and also the loop of Henle. Those parts are majorly responsible for reabsorption of water. All right. And then what will take place now is that since water is reabsorbed, water is conserved, which is point 4. What is conserved in the collecting duct, and the urine will become more concentrated because you're not going to urinate a lot now, you're going to urinate less. And so this also causes less urination, and as a result, the blood will become more diluted and more water will be available to the cells of the body. Because if the cells don't have water or enough water, then you're going to start to feel the effect of dehydration, and then you may lose your life. All right? So you got to be, the cells have to be hydrated to work. And I wanted to make a mention to the way I make my points. Really and truly, there's no need for you to write no long epistle or no long extended paragraphs. You just need to go to the points. They're looking for your points. It's always quality over quantity. It's not the amount you write, but what you write. And also, I would say make sure you have a certain order of flow in terms of the explanation. All right. So don't need to be writing these long paragraphs and then you're trying to figure out where the points are and all that. No, unnecessary. Totally unnecessary. Now, part 2 suggests one way in which Joy can correct the fluid loss she experienced as a result of vomiting. And simply drink some water, drink some fluid, whether it be juice or whatever it is, or just simply drink rehydrating fluids. Like example, Gatorade. All right. And of course, Pedialyte and all those type of stuff. Right. Anything like that. You're right, you get your marks. All right. Part D is explain 2 ways in which plants conserve water. And I have 4 ways right here. One is closing their stomata. Very easy. So if it's losing too much water, the stomata will be closed. And of course, you could explain that this is closed by the guard cells. All right. And this will reduce water escaping by evaporation. Plants also have adopted thick waxy cuticles. Some plants, as a matter of fact, have those thick waxy cuticle. And the waxy cuticle generally, whether it be thick or not, the waxy cuticle creates a barrier against evaporation, and also waxy cuticle is responsible for reflecting light and heat to reduce evaporation. Some leaves are very narrow or small. Smaller leaves or narrow leaves, or even leaflets. For example, like some cacti, right? Leaflets, not really big leaves or defined leaves or well-established leaves. And these now generally have fewer stomata, if any, in some cases. And so, therefore, if you have reduced stomata because of reduced surface area, then naturally you are going to have less evaporation. Some plants also shed their leaves. And the reason for shedding the leaves is to reduce the amount of surface area for evaporation, or simply reducing the amount of stomata that are present on the leaves or plant to cause evaporation. All right, so those are 4 ways that you can have. And that is number 15, and we're end of the test. All right, so again, I wish you luck on the upcoming exams. And for those who are just reviewing for knowledge's sake, thank you for watching as well. And please share with your friends, of course, and I'm looking forward to see you again. Take care. Have a great one.